Welcome to the Photosynthesis Lab. We are going to be playing with plants today. Now, I am going to show you some modifications and some tips for how to do today's lab. One of the things you'll notice when you get to the material list is you'll notice it's a lot shorter than the material list that we have had in the past. So really the emphasis of this lab is on applying the concepts and understanding what's happening behind the scenes, what's happening in our experiment. So it's going to be a little bit more concept heavy than experimental method or technique heavy. So one of the first things that Hands-On Lab wants you to do is to get a paper towel, and they want you to get that paper towel wet. Now I had one of these little spray bottles in a travel case, and what I realized is that I could fill this with some distilled water, and this will make getting my paper towel wet a lot easier. I can get a nice regular covering. It doesn't get sopping wet and start to tear. And the reason why we're getting the paper towel wet is one of the things that we're going to be doing is we're going to be cutting little discs from a spinach leaf, and we don't want those discs to start to dry out. If those discs start to dry out, then our experiment will not be successful. Now, I'm biased. I love plants, and one of the things you may not know is that a leaf is really, really fluffy. There's lots and lots of surface area. There's not a lot of volume. If we look underneath the microscope at a cross-section of this leaf, we would see lots of air spaces. The reason for that is this leaf is all about gas exchange, getting carbon dioxide in, getting rid of oxygen, and, unfortunately, getting rid of water at the same time. So we are going to manipulate the fact that this leaf is really fluffy and is full of air spaces. So in order to do that, we need to cut little discs. We're going to use our hole punch. And when we cut our discs, we want to make sure to cut the fluffy part of the leaf. So if you flip over the leaf, you're going to see a very thick vein. And then you'll see these even thick side veins coming off. Do not cut your disc across any of those veins. We need to get in between the veins where our leaf is really fluffy. Now I've cut a few discs that you can see on the paper towel here. One thing that I noticed is that you have to pull down on this little tab, and the rest of the discs will pop out once you have cut them. So I've cut my little leaf discs. And what I'm going to do is I'm going to fold my paper towel over those leaf discs to keep them nice and damp while I am setting up my treatments. Once you have your treatment set up, one of the things you're going to need to do is you're going to need to fill the air spaces on your leaf. So I'm going to give you a quick demonstration of how we fill the air spaces in our leaf with our different treatments. So if this is a treatment of water, what I'm going to do is I'm going to take my syringe, and these syringes are new, and one of the things that happens when these are new is this black rubber coating is super, super stiff. So my advice to you is to pop this in and out, start to loosen that rubber a little bit so it makes this part of your experiment easier. So we take this portion out of our syringe, so our syringe is empty, and then into our syringe we're going to put our little leaf discs. Be as gentle as you can. You don't want to damage these leaf discs. And you'll notice I'm kind of tapping a little bit on the side, and that'll get all of our leaf discs right down in the bottom of our syringe. We're going to put the stopper in, and this is where we have to be very, very careful. We're going to push out all of the air in our syringe. Try to get it as close to those little leaf discs as we possibly can. Then we're going to submerge our syringe in our container of water, and we're going to pull up a few milliliters. Now, you'll notice I still have a little air bubble. We've got to get rid of that air bubble. So I flip it upside down, and I very gently press the stopper, pressing out all of that air till just water flows out. Now I have a continuous column of water. And what you'll notice is all of those leaf discs are floating near the top. The technique we now have to use to force all of this liquid, to force all of this solution into the air spaces of our disc, is to hold the syringe in between my first and middle finger and thumb. When I hold the syringe in this way, my thumb is pressing tightly against that edge. I now pull back. And what you can see is I've got a little bit of air that has started to move in along the top. That's okay. My vacuum is very tight. Right now I can feel that pressure on my thumb. I can feel the plunger pulling back against my fingers. You don't have to hold yours this long. I'm just doing this for the demonstration. So you pull your plunger back, gently release, and release your thumb. Now, what should happen is your leaf discs should start to sink towards the bottom. If your leaf discs don't sink, just gently press up again. Make sure you've got nothing but water on the inside. Put your thumb over the top and pull that vacuum back again. Release. And they should all fall to the bottom. I suspect the reason why mine are not falling to the bottom is that they are still stuck to the side. So I'm going to try one more time with the vacuum. Press all my air out. Pull deeply back. Release. And most of them are falling to the bottom. I don't actually want to do this too many more times. If I do this too much, I could run into the problem of damaging the discs. So it looks like I've got 2 that have not quite fallen to the bottom, and that's okay. I just need to keep that in mind when I'm analyzing my data. I'm then going to dump this into the cup. And all of the leaf discs should come out as well. If all of your leaf discs do not come out at one time, I've got one little leaf disc, or a few little leaf discs still stuck in there. Just gently replace the plunger. Suck up some more of the water, swish around, and dump again. There we go. Make sure all of your leaf discs are in their treatment. Most of your leaf discs will sink to the bottom. If 1 or 2 are floating, just keep that in mind. We wanted them to all start here at the bottom. So in your experimental setup, you want to make sure that you have cut out at least 40 discs. My big piece of advice is to cut out 45 or maybe even 50 leaf discs, because sometimes these little leaf discs get away from you. Keep that paper towel wet so you're keeping your leaf discs healthy while you are getting them set up. And by getting them set up, what I mean is getting the cup filled with the right solution, getting those air spaces in your leaf disc full of that solution by using the syringe, and then finally getting everything into the treatment setting, whether that's under the light or whether that's in your dark environment. This experiment is going to run for a while. You're going to count the amount of floating discs, And so when we look at this experiment, we want to first identify what is our experimental group and what is our control group. Our experimental group is going to be the group that is getting the independent variable, that is getting that carbon source. It's also going to be the group that is getting either the light or the dark treatment. So we've got two independent variables that we're playing with in this experiment. We're playing with carbon and we're playing around with light. So a big question is, what is our control group? Well, our control group is going to have to be the leaf discs that are in the distilled water because they're missing our independent variable and they are underneath the light source. And the reason why is because we know that photosynthesis requires light in order for those two separate but metabolic pathways to work. We're controlling for everything that we possibly can. We're using 10 discs per cup. We're filling the cups with the same volume of solution. It's hopefully the same person who is doing this entire experiment. So we're holding the researcher constant. Room temperature will be the same. Hopefully any airflow will be the same. The time will be the same. We're holding as many things constant because we want to know that our leaf discs our spinach leaf discs are responding to the carbon source and responding to the light. So let's do a quick review of photosynthesis. So I always talk about how photosynthesis is two separate but connected metabolic pathways. We've got the light reactions and the dark reactions. Overall, we know that our inputs are going to be carbon dioxide and water. We're going to need some sunlight to force carbon dioxide to become glucose. And we'll happen to get some oxygen. But please do not get focused on oxygen. That is not the reason why plants do photosynthesis. They don't care if humans and animals live and breathe. Oxygen is actually a toxic gas and can halt and stop the photosynthesis process. So plants are really trying to get rid of oxygen. They just don't care about us whatsoever. So if you have that point of view, you're going to have to rewrite your brain. Plants don't care about you. They want to eat you for food. You are the fertilizer. They're not making oxygen so you can breathe. It just happens to be a byproduct of the photosynthesis process, a byproduct of these metabolic pathways. So if we have these two separate but connected metabolic pathways, the first pathway is always going to be the light reactions. Now remember, the light reactions are happening in the thylakoid membrane of our chloroplast organelle. Our inputs are going to be water and sunlight. That's all we need. Water is going to give us electrons. Sunlight will get those electrons excited. And once we get those electrons excited, we can get lots of work done. And part of the work that's going to be done in our light reactions is making NADPH and ATP energy. So the first part of photosynthesis is all about using electrons, using the sunlight energy to get electrons excited. And when you get those electrons excited, you can make ATP and NADPH energy molecules. The second part of photosynthesis is our dark reactions, sometimes known as the Calvin cycle. Our inputs for the Calvin cycle will be carbon dioxide, and then we'll need ATP and NADPH energy from our light reactions. Now, the whole point of the metabolic pathway of the dark reactions or Calvin cycle is to force carbon dioxide to become glucose. Carbon dioxide is a super stable molecule. One carbon dioxide molecule can stay in our atmosphere, having an effect on our planet for over 100 years. That molecule is super stable. It doesn't want to become anything. So the amazing thing about the dark reactions is that the plant can turn carbon dioxide, force it to take on all of these hydrogen, force it to take on lots of electrons, and turn it into glucose, turn it into that monosaccharide, that sugar. Now that monosaccharide gets bonded together into polysaccharides like starch or cellulose, and either you eat it directly in the form of a peach or a strawberry or some lettuce or the spinach that we use today, or you run it through a cow or a chicken or a pig and you eat your monosaccharides that way. Either way, you are eating what essentially is stored sunlight, because plants are grabbing all that sunlight energy and eventually storing it in the form of glucose. Now, in the process of the light reactions, taking water and making ATP and NADPH energy, plants will happen to make some oxygen. That oxygen could get grabbed during the dark reactions and stop everything from happening. So plants really want that oxygen to go away. So in our experiment today, what I will be looking for, or what I am hoping you will be understanding is happening inside of those little itty-bitty discs, is you're getting down to an understanding the relationship between the light and the dark reactions. If we have no light, we won't make ATP or NADPH energy. If we can't make these energy molecules, our dark reactions can't happen. We're not going to make any glucose. If we're not doing the light reactions, our plant will not be making oxygen. Now, oxygen, when it's being released, will fill those air spaces in our leaf discs and potentially make them float. If our plant has carbon, some kind of carbon source, it can do the dark reactions, but it's going to need ATP and NADPH energy. So if it's got lots and lots of carbon source, but it has no energy molecules, nothing else can happen. The plant just waits. Nothing else gets released, glucose doesn't get made, we're not making energy molecules, so we're not splitting water and making oxygen, nothing else can happen. So keep that in mind when you're doing exercise 1 today and you're doing the experiment and writing up the results. Come back to this lecture and revisit these details of the light and the dark reactions. Now, the second thing that you will be doing today is kind of an exciting part. Usually, everything is so structured in these labs. We tell you everything to do. But for the second exercise today, you get to play around. You've got the technique now. You know how to cut spinach discs. You know how to fill those air spaces with some kind of solution. Now you get to play around with different variables. So one thing I like to play around with plants is the different colors of light. If I'm growing lettuce, I know that lettuce loves the far red spectrum of wavelengths of the light spectrum. I know that some plants are not going to do so well in blue. If you go to the RVC greenhouse, there's a big LED light in the display window that has, if you look at it, some lights are just blue, some are yellow, some are red. So you can play around with the variable of wavelengths of light by changing the color of light that you're exposing your leaf discs to. You could play around with temperature. I know that some of us tend to have cooler houses or cooler places in our houses, and some of us have warmer places. You could play around with sunlight and actually take this experiment outside on a sunny day and see if that affects your leaf discs any differently. So for exercise 2, you get to pick the independent variable. Pick just one. Keep it simple for this one. And I look forward to seeing what you do and what you change. That to me is really exciting because I know you're very creative and I want to see what kind of creativity you bring to this experiment. Once you've got exercise 1 done, understand what's happening, and are familiar with the techniques. As always, you know how to get a hold of me. You know to email me or stop by my Zoom office hours. My last tip before I leave you today. Our next lab is cell respiration. In order to do cell respiration, we're going to be germinating millet seeds. Millet is a type of grass. So that's going to take a few days. As soon as you are done with this photosynthesis experiment, I would get those millet seeds started. Get them germinating so that it doesn't delay you in completing your cell respiration lab.